This is the last video in the microscopy unit. Previously, we talked about the differences between a light microscope and an electron microscope. However, there are lots of different types of light microscopes. So in this video, we're going to look at some of the main types of light microscopes, talk about how they differ, and why you might choose to use one type of microscopy over another type. Okay, let's get started.
There are many different types of light microscopes, which are all built and function differently from one another. You do not need to understand the inner workings of the individual microscopes for this course. However, you will want to understand why you might want to choose one type of microscope over another one. The four main types of microscopy we'll discuss are bright field, dark field, DiC or phase contrast, and fluorescent microscopy. So let's go ahead and start with bright field, the most standard type of light microscopy. Bright field microscopy is the standard and most common type of microscopy using a compound microscope. This is the type of microscopy you will learn about in lab. One of the keys with bright field microscopes is that most of the cells are fixed, which means they are physically bonded to the slide surface using heat and then stained to improve contrast. This makes the specimen appear a certain color against a bright background. The key to remember is that the fixing and staining process results in dead cells. So this type of microscopy cannot be used to view any movement or functions of living cells. Instead, you use bright field microscopy to examine cell morphology and external structures. In fact, there are different staining procedures used depending on what morphological information you hope to learn. We will discuss those staining protocols as we discuss the structures themselves in the next unit. There are a variety of different dyes you can use to stain cells and different cellular structures, and the different dyes often have different colors. Cell dyes can be divided into two different categories depending on their ionic charge. Positive stains are basic dyes that have a positive charge. Most cell structures have a slightly negative charge to them, so positive staining means the positively charged dye will be attracted to the negative cell and bind to it. This colors the cell itself, as is seen here in this image. Negative staining uses acidic dyes which have a negative charge. These dyes are repelled from the negative cells, resulting in a stained background and clear unstained cells like seen here. When doing staining procedures, some protocols require the use of only one stain, while others require multiple different stains. If one stain is used, that is called simple staining and can be done with either a basic or acidic dye for positive or negative staining. If multiple stains are used, this is called differential staining. This is more common than simple staining because it allows you to differentiate one microbe from another by telling you something about the microbe's morphology. Simple stains will allow you to see the size and shape of an organism, but differential stains provide additional information depending on the stain. The gram stain image that is shown here is one of the most common differential stains used in microbiology. Next, we're going to look at dark field, which functions differently than bright field because instead of the background being illuminated, the actual specimen reflects the light itself. Dark field microscopy uses a special light microscope that actually blocks the majority of light from entering the objective. Instead of light coming from around the specimen and illuminating the background, as we saw in bright field microscopy, dark field causes the light to reflect from the specimen itself, making the specimen appear light in a dark background. This means there's no staining required in dark field microscopy, and the key to remember with no staining is that the organisms can be alive. This is a key difference between dark field and bright field microscopy. You are able to use dark field microscopy to study living cells. However, you cannot learn much about the cell itself besides its shape and size and movement because of the lack of staining. Most often, dark field microscopy is used to view the specific shape of the microbes called spirochetes, which are seen here. These microbes are often so skinny that they cannot be seen clearly after staining in standard bright field microscopy. Two main diseases caused by spirochetes are Lyme disease and syphilis. So often dark field microscopy can be used to examine an STI swab for identification of the Treponema pallidum syphilis-causing spirochete. The next set of microscopy we're going to talk about is DiC and phase contrast microscopy. These are more fancy microscopes, and they're not found in every lab. Phase contrast and DIC, which stands for differential interference contrast, are two different types of microscopy that manipulate the wavelengths of light to produce a more detailed 3D image of cells. This does not require staining and is most often used for eukaryotic cells because it allows for a more high-resolution image of the internal structures as seen in these images, with the top picture showing the phase contrast microscopy and the bottom picture showing DIC. The last type of light microscopy we're going to look at is fluorescent microscopy. Fluorescent microscopy requires the use of a fluorescent dye or protein, but unlike bright field, use of fluorescent dyes does not require killing of the cells. Thus, the cells can be alive or dead in fluorescent microscopy. This type of microscopy takes advantage of the concept of fluorescence. A fluorophore, or fluorescent dye, absorbs a certain wavelength of light, referred to as the excitation wavelength. However, absorbing this light causes the molecule to give off light of a different wavelength, called the emission. For example, the dye in this cell here is absorbing the purple excitation wavelength and then giving off a green glow for the green emission wavelength. That glow is then picked up by the detector and we can see it in the microscope image. Fluorescent microscopy allows you to label specific structures or proteins in the cell with different dyes to view their localization. For example, in this image, the green is actin, the red are microtubules, and the blue is the nucleus. This allows you to visualize the cell cytoskeleton, with the actin typically providing structure under the cell membranes, seen here and here, and the red microtubules spread throughout the cytoplasm, providing structure and transport throughout the cell. Another common use of fluorescent microscopy is to detect the presence of viruses in a sample. Viral detection is done using fluorescent antibody assays. Remember, viruses are too small to be seen in the light microscope. However, if you cause the virus to glow with fluorescence, you can detect its presence just by a glowing dot of light, even if you can't see the virus itself. This assay takes advantage of antibodies, which are small protein molecules that are part of your immune system. Antibodies very specifically bind to a molecule. In this example, the antibodies that we're using bind to the rabies virus. So we have the antibody here that binds and recognizes the protein on the rabies virus, and that antibody is tagged with a fluorescent dye. So if the virus is there, the antibody will bind, and then we'll be able to see the fluorescence from the dye. You mix the antibody with the tissue sample on a microscope slide, and then you view it using the fluorescent microscope. We can see that this brain sample that we're looking at is infected with the rabies virus because we can see the glowing green dots indicating the presence of the antibody with the fluorescence binding to the virus itself, even if we can't see the virus specifically. You should now have a basic understanding of the different types of light microscopes and why you might choose to use one in a given scenario over another.